Hello to all of you. Let's discuss chapter 5 part 3. So in this part I'm going to discuss about carbohydrates and lipids. So we have already discussed about nucleic acids and the proteins, and now we are going to talk about these two macromolecules. So let me go into the slideshow mode, and these are also very important biomolecules. So basically carbohydrates, they are basically a source of energy as well as they are part of our cells also. So carbohydrates, they include sugars and the polymers of sugars, and the simplest carbohydrates are called monosaccharides. So just the way we have discussed that like other biomolecules like nucleic acids and proteins, they are like they are polymers. They're made of subunits, and then these subunits, they are connected to each other by covalent bonds, and they're called peptide bonds in proteins. They're called phosphodiester bonds in the proteins, in the nucleic acids, and they're formed by the dehydration reaction. So similarly, the simplest carbohydrate, they're called monosaccharides, and then these monosaccharides, they are linked to each other by the dehydration reactions, and they're linked to each other by covalent bonds, and those bonds are called glycosidic bonds. So the simplest carbohydrates are monosaccharides, and when two monosaccharides link with each other, they form disaccharides, and when many monosaccharides link with each other and form a long polymer, they are called polysaccharides. So monosaccharides, they basically have the empirical formula. This is the empirical formula of monosaccharides, which means monosaccharides, they have carbon, they have hydrogen, and they have oxygen in them. And this shows the ratio of these elements. So if there is one carbon, then there are two hydrogens, and there is one oxygen. So this is the empirical formula. So empirical formula tells us the ratio of the elements in a in any molecule or in any compound. So in monosaccharides, if there is one carbon, there are two hydrogens and one oxygen. So this shows the ratio of these elements in a monosaccharide. And glucose is the most common monosaccharide. Okay, so and glucose is the most common monosaccharide. And you can see in the glucose there are 6 carbons. If there are 6 carbons, then there are double the hydrogen atoms. There are 12 hydrogen atoms. Sorry. Let me go back. So that is why carbohydrates are called carbohydrates, because if you look at the empirical formula, you will see here. If you look at this, you will see this is water. This is water with carbon. So that is why this is a water with carbon. So that is why they're called carbohydrates. And glucose is the most common monosaccharides. Now the monosaccharides can be classified on the basis of how many carbons they have, or they are classified on the basis of the location of the carbonyl group. So on the basis of how many carbons they have, they can be trioses, they can be pentoses, they can be hexoses. So if they have 3 carbons in them, they are called trioses. If they have 5 carbons in them, they're called pentoses, and if they have 6 carbons in them, they're called hexoses. Then on the basis of the location of the carbonyl group, they are called aldoses, or they're called ketoses. Now if the carbonyl group is at the end of the carbon hydrogen chain, then this group is called aldehyde group, and the sugars are called aldoses. If the carbonyl group is in between the carbon chain, if the carbonyl group is in between the carbon hydrogen chain, then this group is called ketone group, and the sugars are called ketoses. Okay, so on the basis of the on the basis of the presence of the carbonyl group or the location of the carbonyl group, on the basis of the location of the carbonyl group, they are either aldoses or they are ketoses. If the carbonyl group is present at the end of the chain, then this group is called aldehyde group. And if this carbonyl group is present in between the chain, then this group is called ketone group, and the sugars are called ketoses. And if there is a presence of the aldehyde group, then the sugars are called aldoses. Now here are some examples. So in the sugars which have 3 carbons in them, in the trioses, if the carbonyl group is present at the end, then if there are 3 carbons, then this sugar is called glyceraldehyde. 3 carbons, and the carbonyl group is present in between, it is called dihydroxyacetone. Okay, in the pentoses which have 5 carbons, if the aldehyde group is, if there is a presence of the aldehyde group, then the sugar is ribose, and if there is a presence of the ketone group, then the sugar is called ribulose. In hexoses, which have 6 carbons, among the if there is a presence of the aldehyde group, so this sugar, you can see here there are 6 carbons and the carbonyl group is present at the end, so this sugar is a aldose. This sugar is also aldose, and here the carbonyl group is present in between, so the carbonyl group is present in between. This sugar is called ketose. This sugar is called ketose. So these are aldoses, these are ketoses. The group is aldehyde group, the group is ketone group. Now if you look at the structure, this sugar is called glucose, this sugar is called galactose. Okay, so both are hexoses, both are aldoses. Now glucose and galactose, if you look at this structure, they are enantiomers. Okay, so going back to chapter number 4, where we talked about the different kinds of isomers, if you remember, enantiomers are the mirror images of each other. So glucose and galactose, they are mirror images of each other. And this is fructose. Okay, this is fructose, this is a ketose, and this is also a hexose. So if we compare glucose and fructose, you will see that glucose and fructose, they are structure isomers. Okay, because they have, glucose and fructose, they have same molecular formula, but what is the difference? They have different covalent arrangement of the atoms. So they are the structure isomers. Okay, so basically these monosaccharides, they serve as major fuel for cells. So carbohydrates are basically a quick source of energy. Okay, so we are going to talk about fats also. If we compare fats and carbohydrates, both are energy rich, but fats, they have more energy in them, but carbohydrates, they are quick source of energy. Okay, now this is just showing you the structure of glucose. This is the linear form and this is the ring form. So how the ring form is formed? Because this is the linear form of glucose. There are 6 carbons, and basically how a ring form is formed. So basically the carbon number 1, okay, the carbon number 1, which is this carbon forms bond with the oxygen which is linked to carbon number 5. Okay, so this carbon forms a bond with this oxygen which is bound to carbon number 5. So this is carbon number 5, this is carbon number 5, and this is the oxygen which is linked to it. Okay, and this is carbon number 1. So they form together a bond with each other. For them to form together a bond with each other, this carbon needs to break one bond with oxygen. So right now in this situation, the carbon is forming 2 bonds with oxygen. Okay, To satisfy the electronic configuration, this oxygen needs to break one bond with oxygen, and then only it can form bond with this oxygen, and then this oxygen is going to lose a bond with this hydrogen. So to form a bond between this carbon and oxygen, this carbon needs to bond one bond with oxygen, right? Only then it can form a bond with this oxygen, and for this oxygen to form bond with this carbon, it has to break its bond with hydrogen, and then this hydrogen forms bond with this oxygen, and that's the way how ringed form of glucose is formed. Okay, what is a disaccharide? As I said, two monosaccharides linked together by dehydration reaction that results in the formation of a covalent bond, and these bonds are called glycosidic bonds or glycosidic linkages. So you can see here these are two examples of two disaccharides. Maltose is a disaccharide of two glucose molecules, and you can see these two glucose molecules, they together lose a molecule of water. So they're together losing a molecule of water, and that results in the formation of a bond between these two monosaccharides, between these two glucose, and that results in the formation of maltose. Here is sucrose. So this is glucose, this is fructose. So glucose and fructose, they together lose a molecule of water, and that results in the formation of a glycosidic bond between glucose and fructose, and that results in the formation of sucrose. Sucrose is a sugar which we use every day in our daily life. Okay, now polysaccharides. So polysaccharides are polymers of monosaccharides. So they have multiple monosaccharides linked together again by dehydration reactions, and these monosaccharides, they link to each other by glycosidic linkages. And in the polysaccharides, we are going to talk about two different kinds of polysaccharides. They are storage polysaccharides, and there are structure polysaccharides. Now storage polysaccharides are of two kinds. We are going to discuss. One is starch, another is glycogen. So starch is a storage form of polysaccharide in plants, and glycogen is a storage form of polysaccharide in animals. And in starch, we are going to talk about two kinds of starch. One is called amylose, another is called amylopectin. Okay. So you know that potatoes, they are plants, and they are rich source of starch, and starch is a polysaccharide. It's a storage form of polysaccharide. Okay, so you can see here these are two kinds of starch, amylose and amylopectin. So what's the difference between amylose and amylopectin? The difference between amylose and amylopectin is that in the amylopectin there are some in-between branches also. So there are some in-between glucose molecules, they link with each other, and there's a formation of some branches in between. Okay, so amylopectin is somewhat branched and amylose is unbranched. Now glycogen is a storage form of polysaccharide in animals. So like in us, whenever we eat excess amount of sugar, that excess amount of sugar is converted into glycogen, and it is stored in our liver. So it's a storage. So glycogen is a storage form of polysaccharide in animals. So glycogen is also extensively branched. So amylopectin and glycogen both are extensively branched. And the third kind, so these were starch and glycogen. These are storage form of polysaccharides in starches in plants and glycogen in animals. And the next category of polysaccharides we will be discussing will be structural polysaccharides, and cellulose is a typical example of a structural polysaccharide. So in the cellulose, there are basically all these glucose molecules, multiple glucose molecules, they are connected to each other. And an important thing to know about cellulose is that cellulose is also called fiber. So cellulose is present in the cell walls of plant cells, and it is cellulose which makes the plant cells cell wall very hard. So these are the different glucose molecules. They're connected to each other. You can see that. And these glucose molecules in the different polysaccharides, they are also connected to each other by hydrogen bonds. Okay, so they're also connected to each other by hydrogen bonds. So that is why cellulose is very strong molecule. It's all because of the presence. It's all because of the presence of the hydrogen bonds between these cellulose molecules. Okay. So this is all what I said. Okay, so this is amylose, which is unbranched, amylopectin, which is somewhat branched. Then glycogen is extensively branched. This is showing you cellulose molecules. You can see how they are all connected to each other. Okay, glycogen is storage polysaccharide in animals. Glycogen is stored mainly in liver and muscle cells. Cellulose is the major component of tough wall of plant cells. So basically, there are two kinds of glucose. One is alpha form, another is beta form. In starch, the glucose is alpha form, and in cellulose, the form of glucose is beta form. So you can see here, this is the alpha form of glucose, this is the beta form of glucose. So the difference between the alpha and the beta form of glucose is that this OH group, which is linked to the carbon number 1, it is below the plane. It is below the plane of this ring structure. It's below the plane. This OH group is below the plane in the alpha glucose, and it is this hydroxyl group, which is linked to the carbon number 1. It is above the plane. It's above the plane of this ring structure in the beta glucose. So starch has alpha glucose, and cellulose, it's a polymer of beta form of glucose. So this is very important point to remember, that starch is a polymer. Starch is a polymer which is a polymer of alpha form of glucose, and cellulose is a polymer made of beta form of glucose. Okay, something very important to know is enzymes that digest starch by hydrolyzing alpha linkages cannot hydrolyze beta linkages in cellulose. So basically, cellulose is a fiber. So you must be knowing that it's very important to eat fiber. But it's also important to know that we do not have the enzymes to digest this fiber. So then why do we eat the fiber? Because these days it's said by doctors and all that you must eat fiber. So why are we eating fiber? So eating of fiber is very important. It's required for our bowel movements, but otherwise we don't have the enzymes to digest these linkages which are present in the cellulose. So some microbes, they have some enzymes in them which can digest the cellulose. So many like cows and all that who eat all plant-based diets, so they're eating a lot of cellulose. So how do they digest that? Because they have some microbes living in their gut, and those microbes, they have enzymes to digest the cellulose. So we can digest starch, but we cannot digest cellulose. Another and the last kind of polysaccharide we are going to talk about is called chitin. And this chitin is basically this is another structural polysaccharide. It is present in the exoskeleton of arthropods, and it's also present in the cell walls of fungus. And this is the monomer of chitin. And important thing to know about chitin is it has nitrogen in it. Okay, the next category and the last category of biomolecules I'm going to discuss is lipids. Okay, lipids, they are not polymers. They're not typical polymers. So this is exception. Lipids are exception. They are not polymers. They're not made of repetitive subunits. But you will see that the components of which these lipids they are made, they are also made by dehydration reactions. They're also made by dehydration reactions. But those dehydration reactions which lead to loss of molecule of water, that results in the formation of covalent bonds. But lipids are not made of repetitive subunits, so they are not polymers. Another important thing to know about lipids is they are nonpolar in nature, so they do not dissolve in water. And lipids mainly consist of hydrocarbon regions. So basically lipids are made of carbons and hydrogens. And we are going to discuss about 3 different kinds of lipids. We are going to discuss about fats, phospholipids, and the steroids. Okay, so let's start with fats. So what is a fat? So fat molecule is made of 2 things. It's made of glycerol and it is made of fatty acids. Now the important thing to remember here is there's a difference between fatty acid and a fat. So fatty acid, so again there's a word called acid here. So wherever the word you get acid, so something should ring in your mind when you see the word acid, that there is a presence of the carboxyl group, which is COOH group. That is why it's called acid. So fatty acid is a long chain of carbons and hydrogens. And it has a carboxyl group in it. So that's why it's called a fatty acid. And also it has a glycerol. So glycerol is a 3-carbon alcohol. So it has 3 carbons in it, and each carbon is linked to a hydroxyl group. So in the glycerol there are 3 carbons and the 3 hydroxyl groups. And in the fat molecule, 1 glycerol molecule is attached to 3 fatty acids. And fats are also called triglycerides because there is 1 glycerol and 3 fatty acids. So you can see here, this is glycerol. So there are 3 carbons, 1, 2, 3, and to every carbon is attached the hydroxyl group. And here, this is first hydroxyl group, this is second, this is third. So here, this hydroxyl, so here is attached the first fatty acid. So for linking this fatty acid to the hydroxyl group here in the glycerol, so the fatty acid is going to lose OH group and the glycerol is going to lose H. Together they lose a molecule of water. So as I said, these lipids, they are not polymers, but they are made of components, and those components, they are linked with each other by dehydration reaction. So you can see here, they're together losing a molecule of water, and that's resulting in the formation of a ester linkage. So this kind of a bonding is called ester linkage. So same thing happens here on the second hydroxyl group, same thing happens at the third hydroxyl group, and you can see that there is a formation of these ester linkages, and this results in the linking of 3 fatty acids to 1 molecule of glycerol. So this is a fat molecule, or it's also called a triglyceride. Now the fatty acids in a fat molecule, they can be of 2 kinds. They can be saturated fatty acids or they can be unsaturated fatty acids. So the saturated fatty acids are those fatty acids in which the carbons and these carbon atoms, they are linked to each other by single bonds. And unsaturated fatty acids are those in which these carbons, they are linked to each other by double bonds. Because in the saturated fatty acids, the carbons, they are linked to each other by single bonds, they have more hydrogens in them. So that is why they are called they are saturated with, so they are like saturated because carbons are linked to each other by single bonds. So they have more hydrogens on them. So that is why they are called saturated fatty acids, which means like they are saturated with hydrogens. On the other hand, the unsaturated fatty acids, because they have double bonds in them, they have less hydrogens in them. Because they have double bonds in them, they have less hydrogens in them. So that is why they are unsaturated with hydrogen. So that's why they got the name. So saturated fatty acids, single bonds; unsaturated fatty acids, 1 or more double bonds. Okay. If the fat molecule has a saturated fatty acid in it, then that fat is called saturated fat. If the fatty acids are unsaturated, if the fatty acids are unsaturated and when they are joined to glycerol, then the fat molecule is called unsaturated fat. Now, something very important to know about saturated fats is that they are solid at room temperature. But on the other hand, the unsaturated fats, they are liquid at room temperature. So, the best example of saturated fat is butter, and the best example of unsaturated fat is any kind of oil. So you know that oils, they are liquid at room temperature, and butter, which is a saturated fat, it's a solid at room temperature. So why saturated fats are solid at room temperature, and on the other hand unsaturated fats are liquid? The reason is in the bonding. Because saturated fats, they have all single bonds in them. You can see this is glycerol with 3 fatty acids. They all have single bonds in them, and because of the presence of the single bonds, these 3 fatty acids, they can stack on each other, and that's why butter is solid at room temperature. It's all because of the presence of the single bonds. Now this is your unsaturated fat, which is your oils. They are liquid at room temperature because of the presence of the double bonds in the fatty acids, and because of that there's a kink in the structure. So you can see how this fatty acid is bent, and these two are having single bonds. This is the one having double bond. So because of the presence of the double bond, you can see there's a bend in the structure, and because of the bending here, these fatty acids, they cannot stack on each other, and that's why it is not solid, but it is liquid at room temperature. So fats made from saturated fatty acids are called saturated fats and are solid at room temperature. Most animal fats are saturated. Fats made from unsaturated fatty acids are called unsaturated fats or oils and are liquid at room temperature. Plant fats, fish fats are usually unsaturated. Okay, so we know that the food which is rich in saturated fats is not good. So it can basically make us more susceptible or prone to the cardiovascular diseases. So the food which is rich in saturated fats is not good. And similarly, there is a process, there are other kinds of fats which are called trans fats, and they are also not good for our health. And they can also make us more prone to the cardiovascular diseases. Now, what are trans fats? So trans fats are usually formed during the process of there's an industrial process called hydrogenation. So the process of hydrogenation basically means adding of hydrogen atoms. The process of hydrogenation means the adding of the hydrogen atoms. So the hydrogenation is a process of converting unsaturated fats to saturated fats by adding hydrogen. So unsaturated fats, they are liquid at room temperature, and saturated fats, they are solid at room temperature. So why unsaturated fats should be converted into saturated fats? As I said, unsaturated fats are liquid and saturated fats are solid at room temperature. So sometimes some of the industries, they want to change unsaturated fats. They want to change unsaturated fats to saturated fats, which means they want to change liquid fats to solid fats, because liquids are sometimes difficult to transport. So some of the times these companies, these industries, they want to convert unsaturated fats to saturated fats so that they can become solid. And how a unsaturated fat be converted into saturated fat? It can be done only by the addition of hydrogens, because in the unsaturated fats there are double bonds, in the saturated fats there are single bonds. So by the addition of hydrogens, unsaturated fats can be converted into saturated fats, and this process is called hydrogenation. And during this process of hydrogenation, there is the formation of the trans fats. There is a formation of the trans fats, and these trans fats are also bad for our health. So as I said, fats, remember this is very important thing. Fats have the maximum energy in them. If we compare carbohydrates and fats, there is more energy in the fat molecule as compared to carbohydrate, but carbohydrates are a quick source of energy. And in our body, all these fats are stored in the form of adipose tissue, and in the adipose tissue there are nothing but in the adipose tissue there are the fats are stored as triglycerides. Okay, the next category of lipids are called phospholipids. So phospholipids are basically very similar to the triglycerides, but the only difference is in the triglyceride there is one glycerol with 3 fatty acids. In phospholipids there are one glycerol with 2 fatty acids, and instead of the third fatty acid there is the presence of the phosphate group. Now the phosphate group, you already know, that's a charge group. It has negative charge on it. So the two fatty acid chains, they are hydrophobic, but because of the presence of phosphate group instead of the third fatty acid, the phospholipids, they have a charge also on them. So one side of the molecule is polar, on the other side where there are 2 fatty acids, that side is non-polar. So phospholipids, the structure is like this, you can see. So basically this is the glycerol molecule, okay, and here are attached the 2 fatty acids, and on the third one, on the third carbon, instead of the fatty acid there is a phosphate group. So this side of the molecule is polar in nature and this side of the molecule is non-polar in nature. So we can show phospholipids by this structure. You can see these phospholipids, they have a polar head. Okay, so they have a polar head, and this polar head is hydrophilic in nature, and then these are the 2 hydrophobic, and these are the 2 fatty acids. So this side of the molecule is non-polar, or it is hydrophobic in nature. So phospholipids, they have this side hydrophilic and this side hydrophobic. So phospholipid is also called an amphipathic molecule because it shows both the nature. So important thing to know about phospholipids is when the phospholipids, they are added to a glass of water, then they arrange themselves in this configuration. So these phospholipids, they form the biolayer. Okay, they form the bilayer. So this side of the phospholipids is hydrophilic, which means this side can face water, and these are hydrophobic tails. They cannot face water. They are hydrophobic. They are afraid of water. So basically these phospholipids, in the glass of water, they are going to arrange like this, because in the glass of water is every side. So this end of the phospholipid can face water, but this side cannot face water. So if you put the phospholipids in the glass of water where water is everywhere, they are going to arrange like this. They are going to form the bilayer, because in this arrangement these hydrophilic heads, they are facing water, and the hydrophobic tails, they are facing each other, because hydrophobic tails means they are afraid of water, so they cannot face water. So what do? They face each other, and in this arrangement they can protect the exposure of water. So same thing happens for our plasma membrane. So our plasma membrane, which is an external boundary of the cell, it separates the cell from its environment. So if for the cell, in the cytoplasm there is water. So inside the cell there is water, and outside the cell also there is water. So it's just being like in a glass of water. So water is everywhere. So the cell membrane, the plasma membrane, is made of phospholipids, and because there's a water outside as well as there's water inside, these phospholipids, they form a bilayer like this. Okay, the last category of lipids is steroids. So something important to know about steroids is they have 4 carbon rings fused to each other. Okay, and cholesterol is a kind of a steroid. It's present in a plasma membrane, and we will talk in next chapter what's the role of cholesterol in the animal cell membranes. So it's a steroid. So this is the basic structure of steroid. You can see there are 4 carbon rings which are fused to each other, and then there are some functional groups. Okay. So basically, you need to only know that steroids, they have 4 carbon rings fused with each other. So they have 4 carbon rings fused with each other, and then there are some functional groups. Here I would like to remind you about the example of steroids. If you remember from our previous chapter, last chapter, where we were talking about the functional groups, we talked about the structure of estrogen and testosterone. So both are steroid hormones, and you know that both those hormones, they have 4 carbon rings fused with each other, and then there is a presence of some functional groups. So if you remember, we talked about estradiol and testosterone. They are both steroids, both have 4 carbon rings fused to each other, but there is a difference in the presence of some functional groups which make these 2 hormones different from each other, and these 2 hormones make male a male and female a female. Cholesterol is another example of steroids. Okay. So proteins we have already covered. So this is all from this chapter.